Good morning to one and all. Today, we have gathered here to participate in Dr. Y.R. Sarma Memorial Lecture, jointly organized by ICAR-Indian Institute of Spices Research, Kozhikode, and Dr. Y.R. Sarma Memorial Trust. This program is being organized to honor Dr. Y.R. Sarma, former director of ICAR-IISR Kozhikode, and an eminent scientist who worked on the management of diseases of spices and plantation crops for more than 30 years at ICAR-Indian Institute of Spices Research. Dr. Sharma was the chairman for Black Pepper Integrated Pest Management Committee at IPC Indonesia and FAO consultant for Indonesia, Vietnam, and Sri Lanka. He was the network coordinator for the National Network Project on Phytophthora Diseases of Horticultural Crops, that is PhytoNet, funded by ICAR, and was leading several externally funded projects of this institute. The Dr. Y.R. Sarma Memorial Lecture Series started in the year 2017, and our distinguished speakers were Dr. Dilip Kumar from NIST Trivandrum, Dr. Apparao Podale from University of Hyderabad, then Dr. K.R.K. Reddy from Sri Bioaesthetics Hyderabad, and Dr. M.S. Reddy from Auburn University. And today, we have the fifth lecture in the series. Let's begin today's program with the ICR song.
जय जय कृषि परिषद भारत की सुखद प्रतीक हरित भारत की कृषि धन पशुधन मानव जीवन दुग्ध मत्स्य फल यंत्र सुवर्धन वैज्ञानिक विधि नव तकनीके पारिस्थितिकी का संरक्षण शस्य श्यामला छवि भारत की जय जय कृषि परिषद भारत की हिम प्रदेश से सागर तट तक मरुधरती से पूर्वोत्तर तक हर पथ पर है मित्र कृषक की शिक्षा शोध प्रसार सकल तक आशा स्वावलंबित भारत की जय जय कृषि परिषद भारत की जय जय कृषि परिषद भारत की
May I now request Dr. Santosh J. Epen, Principal Scientist and Head, Division of Crop Protection, ICR IISR Kozhikode, and Secretary, Dr. Y.R. Sarma Memorial Trust, to formally welcome the gathering.
Esteemed Chief Guest of the Day, Dr. M.K. Naik, Honorable Vice Chancellor, University of Agriculture and Horticulture Sciences, Dr. Rema, Acting Director of IISR, other dignitaries, contemporaries of Dr. Sharma, colleagues, friends, students, and family members of beloved Sharmaji. A very good morning to all of you. Once again, we are meeting virtually in our desire to pay our respects to Sarmaji. This lecture is the fifth in the series. More than an academic event, it was our humble initiative to remember our Guru and pay our tributes to one of the pioneering plant pathologists in this sector, an untiring scientist, and more than anything else, one of the finest gentlemen we have seen, a true friend of the farming community. When we in Crop Protection Division started this lecture series modestly 5 years ago, on 28th June 2017, we were not sure how this will go ahead. But surprisingly, by next year, his family, close friends rallied behind us, and it has taken the shape of a memorial trust, which was launched on 19th May 2018. And we are glad that the announcement was made by none other than Professor Apparao Podale, Honorable Vice Chancellor of University of Hyderabad. We have formed a board of trustees with the director of IISR as the president, and the registration process is still pending with the registrar of societies. So we started mobilizing funds, and many of his close friends like Dr. K.R. Kennedy is there, and many others have come forward to support this venture. We have several dreams, but COVID has shattered our plans and slowed down our pace. Still, we could continue to organize the lecture series in a virtual mode. And last year, Professor M.S. Reddy delivered the memorial lecture from USA. We are launching the website of Dr. Vayashama Memorial Trust today, and we are proud to present you the 5th lecture in the series by another distinguished plant pathologist today. Yes, distinguished invitees, my dear colleagues and friends, I am happy to introduce and welcome today's chief guest, Dr. M.K. Naik, Honorable Vice Chancellor of University of Agriculture and Horticulture Sciences, Shivamogga, Karnataka. He did his undergraduate and postgraduate studies in US, Bengaluru, and PhD from IRA. Subsequently, he carried out his postdoctoral fellowship on PGNP wilt pathology in ICAR-Indian, Hyderabad. He started his career as a scientist in IISR, Bengaluru, and served there for 7 years. From there, he shifted to US, Harvard, and Raichur, and served in various capacities, such as associate professor, professor and head, dean of PG studies, 28 years of stupendous service in various roles. He was elevated as Vice Chancellor of UHS on 31st March 2018. He is a recipient of several awards and has won prestigious ICAR Bharat Ratna Subramaniam Outstanding Teacher Award. And I am not listing or reading all of them. He has carried out several professional assignments within and outside the country. He guided a number of doctoral and PG students. Dr. Naik is an accomplished teacher, achiever of excellence, voracious writer with more than 190 research papers, 58 book chapters, 11 books, and he has 2 patents to his credit. So he has contributed immensely to fluorescent pseudomonas, another pet area of Sarma ji. And his other areas of research interest were acetobacter, alternaria, fusarium, and of late aflatoxins. There is so much to speak about. Due to paucity of time, I am constrained to restrict myself. On behalf of Dr. Vayashama Memorial Trust and Indian Institute of Spices Research, I have great pleasure in extending a very warm welcome to you, sir, as the chief guest of today's function and to deliver the memorial lecture. Let me welcome our director, Dr. J. Rema, who is presiding over today's function. Hearty welcome to you, madam. Based on our request, many dignitaries have joined this virtual meeting. I find our many of our Sharmaji's former colleagues, friends, close friends, and we are happy to welcome you all to this function. We have with us today his beloved daughters, other family members, several of his farmer friends, students. On behalf of IISR and the Trust, I am extremely happy to welcome you all. I also welcome all my colleagues, research fellows, who have joined online because of the never-ending lockdown here. We look forward to your continued support and cooperation. Let us together fulfill Sharmaji's dreams. Thank you and welcome once again.
Thank you, sir, for extending a warm welcome to one and all. May I now request Dr. J. Rema, Director, ICAR-IISR Kozhikode, to deliver the presidential address.
Distinguished Chief Guest of the Day, Professor Naik, Vice Chancellor, University of Agriculture and Horticulture Sciences, family members of Dr. Sharma, Dr. Sandosh, colleagues from Indian Institute of Spices Research, and students, farmer friends, and ladies and gentlemen. A few minutes back, we heard a glimpse of Dr. Sharma by the comparer, Dr. Praveena, who told us what Sharmaji was and what was his contribution towards the society. Because there is no one here who do not know Sharmaji, and I think after the lead talk, there'll be a launch of the website and more details of Dr. Sharmaji's work would be presented here. But then, for me, I used to think that Dr. Sharma was a great visionary, because all of us know that from the beginning, he was a person who was working on fight of Torah. But later, when everyone was talking, of late, everyone is talking about environmental pollution, then pesticide residues and other things which is harming the society. Way back in 1990 itself, Sharmaji thought of this, and he started, besides his main work on Fight of Torah, he started working on PGPRs. I think today that those work or the foundation stone what he has laid for starting the work on PGPRs has led us today to a great, we have a lot of technologies, green technologies today, which is addressing many of the problems of what we are facing. Because as on date, we all of us know, I'm just talking from angle of spices because Sharmaji has spent most of his time on spices. We have a lot of PGPRs which help us to have safe pesticides and which has been developed from the PGPRs, which the research work what he has initiated. Another interesting aspect what comes to my mind today is like he has laid a foundation stone for this institute from nothing. Maybe the land of we had a land, and along with his team, I would like to remember Dr. Ravindran, Dr. Prem Kumar, and Dr. Sharma, the trios who have developed this infrastructure as what we see today from nothing under the guidance of many of our previous directors. But that is the infrastructure facility what he has laid. But I would also like to remember the most important thing. That is, other than his work on Fight of Torah, once again, I'm going back to PGPRs because that is one research area which is very much needed today. And the foundation stone what he has laid a long time back has taken our research even to the highest official, I cannot say official, the president of India, of this country, and also to the prime minister of this country, maybe in the form of biocapsules for delivering the PGPR. All this would not have happened if 3 decades before, 3 decades ago, if we would not have started this research. So I think we should, all of us should feel proud that Dr. Sharma was with us and he has laid a strong foundation for us as far as research is concerned. Now, for me personally, when I had joined in 1987, I was posted in crop improvement. During those time, Dr. Sharma was working on Fight of Torah, and it was at that time that they have started, they have identified that Piper colubrinum has resistance to Fight of Torah. So the moment I joined, Sharmaji called me and gave me 2 work. One is to do a grafting of Piper colubrinum and Piper nigrum so that Piper colubrinum could be used as a graft. So that was the first research which I have done in this institute, and that was the first of its kind in our country. That idea has come from Sharmaji. So what I wanted to say that we had a foresight, we had a vision, and today this particular technology is being taken up by many of the farmers all over, in all the pepper growing, most of the pepper growing areas where it is suitable. And another thing what I would like to remember at this time is like the last conversation what I had with Dr. Sharma ji was when my son was getting married. It was in 2016, and my son's marriage was on May 18th. I called Sharma ji and asked him, I just invited him. Then what he said is, Rama, I am there. That's what he told me. But he did not come for the wedding. But on 19th morning, the next day, I heard the sad news that Dr. Sharma is not with us. But I would say that whatever contributions he has made to this country through our institute and through various, wherever he was in, through various forums, have taken our country to great heights, especially in the field of spices. And he lives among the people through his research publications, through the students he has guided, through the friends, and mostly through the farmers. I would, at this instant, I would also like to remember a farmer who has come and met me a few months back. He didn't come to meet me. Actually, he walked into my room by mistake. But then the only person he was inquiring about was Dr. Sharma. So that particular small, even though it was just a casual inquiry, but I was just thinking how much people remember Dr. Sharma even today. And he was telling how he was helping them to solve the problems in black pepper. That time, the wilt was a serious disease. So that is what I was just thinking. I think I will not prolong my talk because we have to listen to Dr. Naik. And once again, with all paying all due respect to Dr. Sharma, I would like to just remember him at this occasion. Thank you, friends.
Thank you, ma'am, for presiding over the function and your presidential remarks. Next, we have the launch of Y.R. Sharma Memorial Trust website. We request our chief guest to launch the website. Thank you, sir. We now have the 5th Dr. Y.R. Sharma Memorial Lecture on Crop Protection During COVID Pandemic with special reference to aflatoxins by Professor M.K. Nayak, Vice Chancellor, University of Agricultural and Horticultural Sciences, Shivamogga, Karnataka. Dr. Nayak started his scientific career as an ARS scientist at IISR Bengaluru. Later, he joined UAS Dharwad and Raichur to serve in various capacities, such as Associate Professor, Professor and Head, University Head, and Dean of PG Studies. He is a recipient of several national and international awards and recognitions. Like few of them, like ICR Bharat Ratna Subramania Outstanding Teacher Award, then Fulbright Fellowship by USA, etc. He is a fellow of 6 professional societies. Dr. Nayak and his team led about 22 externally funded projects on PGPR, biological control, soil-borne pathogens, aflatoxin contamination, food safety, etc., and IDM schedules. An accomplished teacher, achiever of excellence, and a voracious writer with more than 190 research papers, 58 book chapters, and 11 books, with 2 patents filed to his credit. Presently serving as Vice Chancellor of the University of Agriculture and Horticultural Sciences, Shimoga, Karnataka from 2018 onwards. We now request Dr. Nayak to deliver the lecture.
Am I audible?
Yes, sir.
Yes, sir.
Okay.
Yes, sir.
So, very good morning to all of you. Most respected Director of the Indian Institute of Spice Research, Dr. Sarma, Dr. Santosh, the head of the Crop Protection Division, all the faculty members of the Indian Institute of Spice Research, most esteemed family members of Dr. Y.R. Sharmaji. I see his daughter, below daughter sitting there. All the distinguished plant pathologists, other scientists, fellow colleagues, student friends, and those fellow members who have joined Dr. Sharmaji's memorial lecture, very admirers of Dr. Sharmaji, and my student friends. Thank you, Dr. Sarma, the Director of Indian Institute of Spice Research, for giving me an opportunity to be here, sharing the memory of Dr. Sharmaji. As I know him, probably I know him back in 1986, since my IISR days when I was a PhD student there. I know Dr. Y.R. Sharmaji, a very distinguished and dedicated plant pathologist who spent almost 3 decades working on several aspects, particularly spices were very near and dear to his heart. Spices like black pepper, ginger, turmeric, cardamom. He also worked on cashew, cocoa, in fact, a new crop like vanilla. So he was, I think, as I know him, a very simple soul, very humble man, very versatile, most admired by many of his fellow colleagues of plant pathology and rest of the agricultural scientists, both in ICAR and university system. I remember he was the first to have initiated the first international plant PGPR conference was held in Calicut. Dr. M.S. Reddy, Professor M.S. Reddy, the president of the founder president of the Asian PGPR knows it. That was the first PGPR international conference held. And he is very eminent in the sense he had such a great vision so far for his institution. So I would rather consider him as an institutional builder with so much of vision. And today, if Indian Institute of Spice Research, if it is visible on the Indian map, I am sure along with the other directors of the present, and it was Dr. Sharma and his team, and the faculty members are responsible for the dream, and also the Department of Plant Pathology, where the Crop Protection Division. Very accomplished division today, and one would feel proud of it. Absolutely, all these are his contributions. And I remember along with him, I attended many PGPR conferences, which was so near and dear to him. In the last meeting that I met him at Vietnam, where we attended the PGPR. In fact, I joined with him. We traveled together. He was, so much concerned about the PGPRs to be promoted and upscaled and used in India. And he also gave a roadmap during his presentation. Even at Vietnam, as I find him as an FAO consultant, especially for spices, he is most sought after by the farmers, entrepreneurs there at Vietnam, so also in India. So let me pay a rich tribute to the great man, very distinguished personality, for all the contribution that he has made, for the dedication and the service that he has rendered to the plant pathology. Probably the institute and the trust, what we have launched today, I'm sure, which has come with lots and lots of dreams, and his dreams will be realized in the days to come. I'm not sure how the committee elected me to present a topic that to address the memorial address of Dr. Sharma ji. I am too small to make a presentation on a too tall person like Sharma ji. Yet, I will only try to come up to the expectations of you people. So today, while thanking the director and the faculty of the Indian Institute of Spice Research, I have decided to present a lecture on crop protection during COVID pandemic with special reference to aflatoxins. Obviously, this is a tribute that we pay to the great man, Dr. Sharma ji. And the reason that I chose was, we are passing through a tough time in this world, not only in India, globally. Obviously, as we know that the population of India, not only India, globally, today we started with almost 1 billion in 1800, and the second billion we cross just in another 130 years. And the third billion took less than 30 years. And the fourth billion, you look at here, the fifth billion came in 1987, and the seventh billion came in 2010. It is estimated that we will be around 9.8 billion by 2050. And about 0.87 billion are having insufficient food consumption. 15 countries are facing high hunger level. And India being the second most populated country, ICAR estimate says that by 2030, we need to have 345 million tons. While we are just trying to recover and make a substantial gain in poverty alleviation, we have lost the touch right now because of the COVID, which has taken back for a few more years. World today has about 176 million cases with a death toll of 3.81 million, and India nearly 30 million cases with about 0.37 million death toll. So that is the picture before us today. So if you look at the hunger map, with a population of 1,352 million, almost 226 million have insufficient food in India. As you can see here, this particular yellowish area indicating moderately low insufficient food, whereas these two intensive yellow color indication of moderately high, that means at least 20 to 30% insufficient food consumption. So we have almost 17% of acute malnutrition and 34.7% chronic malnutrition. Needless to say that Indian women, most of them have anemic, at least 40%. And then the children have a protein calorie malnutrition. That is the background today we have. We have entered into some kind of paradoxical situation. No doubt today we have 5 times, not 5, rather 304 means 6 times increase in food production, 7 times in horticulture. Milk, we are first in the world. Egg production 27 times, and probably it is a proud feeling that the National Food Security Act enacted in 2013, when we presented in the parliament, highest form of the democratic country like India, that we promised that we will be able to feed 80 crore people through public distribution system. That is the biggest courage that we have. And we are now the largest exporter of rice. All these are good points. But fact remains the same, that we stand in the Human Development Index at 130 out of 189 countries. Global Hunger Index, not a healthy statistics, 102 out of 190. Below poverty line population of 26%. Probably the COVID has again pushed us back. Another 100 million globally are pushed back to the poverty. So the middle class of 32 million is shrinking because of COVID. And in fact, 60% of the global rise in the poverty is also contributed by India. Let us also remember that we cannot build a very developed India on the shoulders of a stunted and wasted children, or in the presence of a larger number of unemployed youth. This is the kind of irony of the story, paradoxical situation. So as we are passing through the pandemics, back almost 100 years back, Asian flu with 1.1 million death toll. On 70s, we had, Hong Kong flu, HIV, AIDS, almost 32 billion. Today, 3.76 million, exactly after 100 years of Spanish flu. That is the major pandemics. Where are we now? Yeah, an unknown virus emerged in 2019 November, and the pandemic unfolded, accelerated in the stage 3. Today we are facing a multifaceted problem of medicine, problem of ventilator, problem of hospital, problem of bed. Pandemic has a multifaceted dimensions today, and medically we are trying to counteract with the vaccination. At least 25 crore today have been vaccinated. But economically, it has decreased more than 4% of the GDP, almost about 100 million people living in extreme poverty due to the crisis. Again, it is a contribution of, what you call pandemic. 9% of the global working hours have been lost. India lost around 10 crore jobs. So this is where we have been pushed back. So the COVID-19, immediately we made an unprepared lockdown last year, March 23rd. So that has deepened, also this year, deepened our inequalities, leading to economic recession, and the supply chain was wholly disturbed. So changes were seen in production, price, and environment. This is how the Committee on the World Food Security has enlisted, leading to increased poverty and food insecurity. So the forecast for the next 5 years of our GDP, last year this was the position. This year is still very pathetic, and next year at best it could be 1.45, at worst it could be 0.45, and traveling through the next 5 years, 24, 25, maybe time taken. At best we could reach 7.45. At worst, it could be 1.45, strong or weak positions. It has not only impacted the society, but naturally agriculture. So the peak harvest affected, no procurement, reverse migration from urban area to rural area, inadequate price and market, the supply chain disruption. Most affected are perishable commodities, and the cash flow also stopped. But the only ray of hope was agriculture did give a ray of hope, in the sense we did not have empty shelves. We did not have a frightening moment because our godowns were full, our fields were green. In spite of COVID, farmers took it as a challenge, and the agriculture sector showed positive growth. That was the best part of the COVID. So we had 2 waves, and the experts anticipate the third wave. But the first wave, of course, we went with unpreparedness. Of course, we did not have even PPP kits. Positivity rate was there, quarantine was the problem, masking, sanitation were the major features. But the second wave characteristically enter the disaster, enter into homes of villages. Migrant labors, of course, affected, medicine, ventilation, oxygen were conspicuous in the absence. That's what we faced. And more so, plant pathologists, probably medical mycologists would agree. The black, white, and the yellow fungi, many more, erupted with high positivity rate, and mortality rate increased, and expenditure obviously enhanced. So agriculture has been affected by both the waves. One of the quick note is that there was a strong association between the food insecurity and the farm size. That means larger the size, they were better secured. Smaller the size, less secure. That means in India, which is a paradise for almost 80% of the landholders of small farmers, marginal farmers. So that is an important point. And in fact, a local survey indicated in our own state, 10% could not harvest, 60% harvested but loss of yield, 25% preferred to store, 12% still trying to sell their produce. So these were the effect of second waves. Borrowing now has become, crop loan repayment has become, because continuously affected delivery of agriculture credit is the problem now. Farmers are approaching money lenders. That is a very problematic. And in fact, informal money lenders are asking collateral security. This has an adverse effect on agriculture. So we have been listening black fungus, white fungus, yellow fungus in the newspaper. All this is mucor fungus, rhizopus, and white fungus, candida. Can also see the symptoms. And this is how the black fungus, especially affecting the nose and the eyes, very terrible. Once it infects, cannot be got ridden easily unless there is a surgery. And of course, aspergillus, and many of these are troubling in the lung infection and aspergillosis and pulmonary infection. These are the problems that we are facing every day. Some of these, I think, plant pathologists, particularly fungal pathologists, probably have to sit and identify. These are some of those opportunistic fungi. No, they are very weak parasites. Saprophytically can grow on a wounded fruits or decaying matter, but then sporulate very quickly. And today, although they were so easily identified in our plant pathological commodities like fruits or cereals or vegetables, especially those which are harvested after harvest, very weak ones. But they are becoming very prominent today, and medical mycologists are also concerned whether there is they are coming together. And we see newspaper yesterday also somebody saying that it is not only on the immunocompromised patient, even on healthy patients. So that's what I think that makes the toughest job. And I think this is an angio-invasive disease, mucormycosis, including rhizopus, rhizomucor. Today, the prevalence is almost 0.14 cases per 1,000, 80 times the prevalence more than the developed countries. And very characteristically seen in immunocompromised patients where there's a diabetes or concomitantly use steroids exceedingly. So the risk factor is involved in such of those patients. So we call it as a black fungus colloquially. So these are the ones involved, otherwise they are not black, in fact, but with the infection on the face manifesting into black color. So this is how you can see the conidia and the conidiophore. As you see, this is the white fungus we call candida and aspergillosis infecting. They are basically allergic fungus, prevalent very much in the air, but also sporulating quickly. India being a tropical country, that is the reason why I have chosen aspergillus today. They can also bring lung infection. So the fungus normally, they live on the skin and inside the body, probably even on the throat, guts and vagina. So that's why the COVID, the patients are at risk, not only COVID, even other patients are also. And the black fungus, the infection results in tissue damage. That's why it looks black. And the yellow fungus rarely visible. Only one case detected in Ghaziabad. Otherwise, the rise in the number of COVID patients in the second wave coinciding with this particular infections are very important for research, not only mycologists, even pathologists of both, medical mycologists as well as fungal pathologists. So aspergillosis, this is what it can also lead to aflatoxin. But in the COVID patient, wherever it can also bring acute respiratory distress, even creating lung cavitations, and pulmonary aspergillosis is common. So they can also invade subacutely the pulmonary aspergillosis. That's why the patients with COVID are more easily vulnerable for this. So as it has affected the agriculture, naturally it has also affected the crop protection. Basically, the plant protection products, whether it's a bio or chemical or botanical, that has been affected. Shortage of labor has affected our protection, whether it is a spray or any other interventions. Financial constraint has affected our protection interventions. Equipment shortage during lockdown. Certification program, the training has been affected. Active ingredient and authorization process has been affected. And even public funding for universities and institutes have been also affected, as you can see in our system. Some of the key issues the International Plant Protection Committee has given is goes back to our age-old proverb: prevention is always better than cure. Phytosanitary certificates, because viruses do not bring passports. So cross-border, we need to have phytosanitary certificates. Risk for the inspectors, because they have to deal with some of these inspecting consignments. And phytosanitary certificate consignment is also delayed. Emergency situations, protecting plant health, food security, because plant health protection is directly linked to food security. Probably pest outbreak, whatever we have seen in these during this pandemic, we can only say pay a little now or pay a lot more later. So health has become a global issue for not only India, even for international community. So the crop specific advisory for crop protection and farm management services is now digitalized. We are talking about digitalized technologies, smartphones, e-clinics. In our own university last year and this year, we have been conducting virtual farmers advice. Yesterday, we conducted virtual farmer field school. It has its own demerits. So the pandemic definitely has affected the crop protection. As we move on, these are agricultural scenarios. So we were food deficient. Today we are. From food security, we have turned into nutritional security, or food safety is the issue now. That is where aflatoxin comes into play. Food import, we are now exporting. That is a healthy sign. From green, we are talking about gene revolution. And as you see during the pandemic, a lot of things were locked down. Primary agriculture production cannot help the farmers. Naturally, you will have to have a shelf life, extended life, value addition, processing, and extending the shelf life was the basic reason under lockdown. Subsistence farming to commercial farming. And we can also see during pandemic, all people were asking about ginkgo, vitamin C, minerals. So all that we need to convert the starch into proteins, into vitamins, if we have to survive the biosecurity. And we need to, that's why we need to diversify our crops, probably from chemical farming, as Dr. Y.R. Sharma ji was friendly, eco-friendly microbial farming was advocated by him. That is what is requested. So we are now talking about the safety of food, safe food for healthy tomorrow. So as you talk on that, there are plenty of issues. One of the issues that we can discuss today, because what we need is a healthy food, safety food, quality food, not only during pandemic. All that is because of, we have hazard analysis critical control point, which can be a global tool to enhance the buyer's confidence. So that is why I chose this particular topic of mycotoxins. Of course, they are very secondary fungal metabolites, cause all pathological and undesirable responses. And this is and there are as many as 20,000 to 3 lakh mycotoxins, although 70,000 have been recognized. 500 are identified by the Council of Agriculture and Science and Technology. And most of them are stable at high temperature, not easily destroyed by the processing. That is our major problem. Aflatoxin B1 and M1, zearalenone, and ochratoxin, lysogenic acid. This is the one which is also used in preparation of LSD drugs, petulin. So these are some of the mycotoxins. These are some of the commodities where you can see in oilseed crop, cereals, aflatoxin is dominating, especially groundnut, legumes, ochratoxin, zearalenone is Fusarium dosium causing on corn, wheat, and barley, especially on cereals. Again, trichothiocin on corn and wheat, citrinin, petulin, the ergotoxin, what Claviceps purpurea, the one which is used by our modern young men and women to open their brain, that the one which causes also gangrene and can be fatal. They are also being used as a drug, particularly to induce miscarriages. So these are some of the mycotoxin producing fungi. So most importantly, they have restricted our trade, and health-wise, they are carcinogenic. They can cause liver cancer, and they also avoid the absorption of nutrition in children. That is why children are also more affected, and it enhances the vulnerability for HIV. So some of the current food safety authority, the limits fixed: cereals and products 15 microgram, and ready to eat food 10 microgram, oilseed 15, and milk 0.5. There are some severe restrictions: wheat and barley 20, petulin 50. So even these are some of the restrictions. You can see United States has restricted 20. European is quite conservative, at least 5 in milk. European Union 10, India of course 20. So these are the limits for mycotoxins in dairy feeds, ranging from 0.5, and fuminosin, another one, Fusarium moniliforme producing. And this is the limit for aflatoxin M1 in milk, ranging from almost 0.02 to 0.5. Various countries like United States, European Union, Australia, and India has a 0.5. So in aflatoxins, of course, the term has been derived because of two combination of Aspergillus and Flavus species and the genus, very naturally occurring and affecting the food production, processing, and transport. These are some of the derivatives, B1, B2, G1, G2, as in the fluorescence, they emit such color and they damage the liver. They're carcinogenic and mutagenic and immunosuppressive, and particularly at a COVID time, they could be very dangerous. So you find them, they are very ubiquitous, present in the soil, rhizosphere, on the upper surface, dead and decaying organic substrates, very easily visible. So the history goes back to 1960. We call it as X disease without knowing the cause. That was due to the infestation by the chicks fed by the groundnut infected with these aflatoxin fungus. Almost so many birds died. That was the X, they call it as X disease. Today, Food and Agriculture Organization has anticipated The survey says as much as 25% of the world food crops are likely to be affected by mycotoxins, both pre-harvest, post-harvest, storage, and processing also. So threat to human health, and they can also bring in plant health like pre-emergent decaying. International trade is most affected. They can bring in the deterioration of the grain as well as fruit quality. So sometimes we have acute aflatoxicosis and a chronic one also. Chronic is can bring in liver cirrhosis. So this is how you can see the contaminated animals giving a scaling of the skin and dwarfing and stunting of the chick fed with certain feed. And you can also see the bloating of the liver. These are some of the manifestation of the symptoms. So this is aspergillus, also called as black mold, as I told you. So produces toxins like varied derivatives like aflatoxin, ochratoxin, fumigatin. So they are all recognized by their yellow, green, and blue and green different colors of molds. So mycologically, they belong to recall they are ascomycetes in the perfect stage, order Uroshiales. Asexual reproduction is by producing conidia in chains on the secondary and the pterygmata. I can see here primary pterygmata produced, conidia produced on vesicle, and that is called pterygmata, and then the secondary pterygmata. So the hyphal cell branches and produces conidiophores over the foot cell. So they produce innumerable conidia. Tropical India being a tropical country, this is the electron microscopic picture. There you can see here the vesicle portion, and then you can also see here how they are basipetal. That means the youngest ones are produced at the base, basipetal manner of this. This is the pure culture of Aspergillus flavus culture, and this is how you can see the sclerotia, the dimensions, and then vesicle formation. They can rupture and release conidia. So this is these are several species. You can see flavus and parasiticus only produce aflatoxin. Of course, fumigatus, Ocrasis niger, fortunately do not. So perfect stage is called as Urosium. So these are the derivatives in B1 and B2, G1 and G2. This is in milk. So these are the chemical structures. So physicochemical properties of aflatoxin, you can see the molecular formula of B1, B2, and that is the molecular weight highest in G2, and the melting point, you can see here over 250 degree, and that is the emission of fluorescence at this nanometer. So the very simple life cycle, they have conidial formation, and then you find them saprophytic growth. The inoculum can be blown by wind, and the secondary spread is very easily visible. This is how the cycle continues. So these are some of the products in India has been worked out: cereals, oil seeds, then vegetables. You can also see them infested groundnut pods, maize, fig fruits, and chili fruits, and milk also. So economically, it has impacted. Of course, we have history in Thailand. In 70s, they have lost about $50 million. And corn in Southeast Asia, several $100 million. And Indian exports of peanut meal, there is a huge loss in 70s and 80s. Spain has detained a few of the consignments of chili. So that has been recorded. So the impact that is across the groundnut value chain, the pre-harvest, you can see starting from the ground itself, then it enters into the poor storage and transportation. You can see them continued. Flow of aflatoxin in food chain, you can see in livestock feed, even the groundnut leguminous product, and then local market where it will be rejected. It's not only not only for export, even domestic consumption also, we cannot take a risk of eating them. So we can't even think of, we can send the better quality outside and we can eat the next quality. No, we can't take any chance here. So the global burden on aflatoxin is estimated 5 billion and up to 25% of the liver cancer is attributed to aflatoxin. So that can always disrupt the trade also. Some of the regulatory actions, sanitary and phytosanitary measures, and some standards have been fixed strictly, but still we have not been able to meet some of these standards. As you can see here, I have chosen 3 crops: chili, which is a spice crop, dear and dear to our Y.R. Sharmaji; groundnut, of course, a very important crop, and in oil seeds; maize, probably aflatoxin is badly affected in maize also. You can see here how the fungal inoculum can infect. This is the, they can easily come and occupy. They are very weak and saprophytes, but sporulation is very quick in humid atmosphere. You can see them very quickly. So this is the newspaper report a couple of years back in Raichur. All these farmers where there is a chili is abundantly produced, and to save the crop and keep the good to get a remunerative prices, they actually kept in cold storage. The owner of the cold storage, with the understanding, kept them in the cold storage. But because of the electricity failures, you can see the large, several tons of them infected with aspergillus, ending in aflatoxin, and farmers are asking compensation. This is how it can, when they are not properly dried and moisture content is not maintained, can always affect. You can see here the infected fruits, those which are near to the grounds, are easily infected, and of course in the heaps also, because always the inoculum is present in the upper surface, and they can, the way we dry up the fruits are after harvest, what you can see here, they can also chance of infection if they are not dried properly. So some of the procedures of phytosanitary issues are involved here. Not moving. Yeah. So most important in aflatoxin is to have a risk-prone areas. One of my student, Guru Prasad, who did his PhD on aflatoxin contamination, made a survey in 4 states. So the survey has indicated, you can see here, incidence of aflatoxin sampling. You could see almost, of course, 0 to 10 in most of the cases, but there was in Mehboob Nagar and other adjoining areas of Telangana, there was 101 to 112 detection microgram per kg. So this is what we can see. This is how the risk-prone areas in chili. So in groundnut, this is, you can see the kernel infected, pods infected, and in groundnut, this is how they are heaping. This method of heaping of groundnut pods after harvest can lead to moisture and fungal growth, whereas healthy groundnut pods, if they are dried to the extent, they can always resist. But otherwise, this is a groundnut pods kept for drying after harvest. This will have a less chance. So means of field contamination, and we can, the groundnut, when it was surveyed, the aflatoxin contamination risk-prone areas, that's what you can see in the adjoining areas of Karnataka and Andhra Pradesh, and also Chitradurga, which is a dry region. We could find 31 to 40 microgram, and again in Kurnool. So these are some of the locations that we can identify. So in maize, there also survey was done wherever the crops are opened, very easily vulnerable. And you can see here, survey done, the sampling. You can see some of the districts in Karnataka, Shimoga, and then Koppal, this is in northern Karnataka. And then Bellary showed maximum infection, where it is a very good area for a large area is grown in maize. You could find in Haveri also. So the GIS map. So similarly, mycotoxin containing maize storage, we could see in the storage, this is after storage, we could see the storage 26 to 50% in our own Shimoga district and Davangere, and around 51 to 55 percentage of samples were not to have infection. Remember, mere presence of fungus does not indicate elaboration of toxin. It has to be detected by, detection techniques, but otherwise it's a mere presence. But that is the data showing. So there are a good many methods, but most of the methods are highly, just PLC, and then you are in indirect ELISA. Those are in specialized laboratories. But one of the culture-based techniques identified is on coconut cream agar. You can see here, in a low-cost laboratory, one can identify the presence of aflatoxin wherever there is a presence of such kind of beige ring around here. That indicates the presence of aflatoxin, whereas the other two do not have that one. And you can, this is when you observe under 365 nanometer. When you reverse on the Petri plate, you can see here the reverse side, the presence of this is a positive, the either side they are negative. So you can, when you put it under long wave of ultraviolet 365 nanometer, there is a emitting of blue fluorescence. That is the indication of aflatoxin presence, whereas the negative isolate will not have that. So estimation, as I told you, there are several techniques. So molecular characterization is also one of the important techniques which can be used. There are, as you know, there are 4 genes essential for aflatoxin biosynthesis: NOR gene 1 and VER gene, OMT1 and APA gene, which is a regulatory protein. So these are some of the genes used for identification of aflatoxin fungi using RT-PCR technique, APA2 specific ones. So they have their own steps in biosynthesis of aflatoxin using polyketide, then to neurosolonic acid. And similarly, you can see here aflatoxin, this is in milk, where it is a versicolorine, which has got this kind of biosynthetic pathway. And this is where these have been used for identification of, you can characterize whether they are toxigenic or antitoxigenic. This is in, they were observed in Chile, some of the isolates when we observe, you can see here the presence of this 10 kb region is indication of toxigenic, whereas in atoxigenic it is missing. Similarly in groundnut, you can see here the atoxigenic as well as toxigenic differentiation. So for all these genes, for example, AFLR gene amplified at 900 BP, and AFLJ gene, which was amplicon, was at 450 BP. Similarly, NORA gene was amplified at 400 BP and VER genes at 400 BP. So this is how we could not find much of the variation in this one. You can find them into 2 clusters. This is from Mandya region. Rest of them under 1 cluster. But what is important, some of these aflatoxin producing isolates we have, after sequencing our DNA, we have deposited in NCBI bank. So these are some of the isolates which have been deposited, and you can see aspergillus flavors deposition. So what is important here is when we compare the PCR and ELISA methods, you can see here all these Chile isolates were more or less found positive, but they produce aflatoxin in micrograms. Sorry, it is missing here, microgram per kg, above 2000 microgram. And in groundnut, we could also see isolates producing 1.9, 2.1, but there is a presence. So this is what, even in spite of the presence of them, there was elaboration of aflatoxin. There was no elaboration here. And this particular isolate missing, but still there is an elaboration. So some of these isolates like this, almost all these genes are missing. We can call it as atoxigenic, absolutely matching. They did not elaborate toxin. So variability is one of those important factors in aflatoxin fungus. As you see, they vary in colors, they produce in different margins, growth, and also rate of growth. So that's what we could not distinguish, but then we grouped them into fastest growing, and then mycelial color ranging from dark to greenish, and then sporulation. Most of them being in a tropical climate, they go for excellent sporulation. That is how they are easily available for infection. So this is aflatoxin B1 when it was estimated in 16 isolates by our own laboratory. As you can see here, in the ecosystem, we could find, this is the passport data of the isolates, we could find the maximum of 1652 microgram per kg elaborated here, and 1081 in one of the isolate from Sindhanur, and the least one of 6.6 again in Bellary. So that itself indicates coexistence of both non-toxigenic as well as toxigenic, but then the elaboration requires those presence of these genes. Yeah. So 59 isolates in another study conducted by our own laboratory. Most of these isolates showed avirulent, nearly 20 of them showed low toxigenic, and then moderately toxigenic based on the quantity, and then toxigenic, there was highly toxigenic, almost 3035 microgram, so about 3000. So this is how in another study, 65 isolates we could find, this is from almost ground at 0, and then some of the isolates very low, mostly avirulent as per our classification. And this is where you can see moderately toxigenic and one isolate highly toxigenic. It is important because after all, the sexuality generates diversity. In aflatoxin fungi, this is the one paper where globally they have taken the isolates from different continents of America, then Africa, Argentina, Australia, and India from Karnataka. At the global scale, they worked out the evidence for the such variation of chemotypic diversity. So this was the work done by the team led by Ignazio Carbone. What it indicates is the mating types, the sexuality can generate the potential of toxin. Of course, in aflatoxin fungus or in aspergillus, we know back in 1950, I think Ponte discovered parasexuality. But then other than that, sexual and asexual and other mechanisms of variability also contributes. So that is how we find the chemotypic diversity. So most importantly, these are the predisposing conditions. See, the most important is presence of fungus in the soil and air. This nobody can prevent, but one can reduce through sanitation. Susceptible cultivars after harvest, then drought stress, terminal drought stress at the time of harvest, particularly for groundnut, is very important. That's where what crucial is. And microflora computation, that's why today we have atoxigenic and toxigenic used as bioagent, atoxigenic ones. Soil temperature, moisture content, mechanical injury, which can, and insect damage. That is how this is one of the important parameters to be noted for management. And premature death of the crop can also, these can come and occupy. So moisture, you can see in chili, one of the experiments conducted, when you dry it below 8%, absolutely no sporulation, no grading at all. But anything above 13 or 27, varied sporulation was observed. So this is one of the important elegant technique, very Fusarium specific, this is Aspergillus specific media, where the soil, the dried and processed one, if you just put it on the media, can estimate the population very easily. So we undertook in some 4 states, what is the extent of population of per CFU per gram of soil. So it ranged in different districts, 510 CFU per gram of soil in Mahbubnagar, Telangana, to as low as 225 in Solapur, Maharashtra. So this is a very important factor which is present in the soil. Soil buildup of inoculum can always play a very crucial role. This is where you can see it varies, but the sanitation can always play an important role here. If you take away all these important inoculum, then it's possible. So we also made amendments using neem cake, fungicides, bioagents, to see the Aspergillus population. As you can see, before sowing, the population was 3.69, and at the end of 90 days, it has come down 52% of the destruction of inoculum with the use of these amendments. So that resulted almost in 0% of aflatoxin. So this is an important step for management, as you can use the amendments like neem cake application. That the most important is especially T6, using not T6, actually T4. That is the most important. 72% wrongly labeled here. 72% decrease, which indicates neem cake as well as consortium of Trichoderma and Pseudomonas use can bring down the inoculum load. This is in chili rhizosphere. You can see here the maximum destruction of inoculum is about 70 to 80%. So at the end of 90 days, 120 days after planting, so which has also brought down the fruit infection as well as aflatoxin contamination. Important factor. So good agricultural practices, what ICAR-ISR has advocated, very important. We have a project funded by ICAR-ISR which helped us to identify this particular seed treatment. You can see here 65% decrease of inoculum for 4.18 to 3.11 after harvest was possible because of good treatments like seed treatment and application of consortium of Pseudomonas as well as neem cake application. Apart from that, good agricultural practices like castor was used as a trap crop. Gypsum application for pod development in groundnut was very crucial. This is again in good agricultural practice. You can see the amendment has brought down the inoculum load from 3.04 10 to 2 to 2.06. Very important factor. So based on some of these prior studies, we took up management of aflatoxin contamination. These are the pre-harvest good agricultural practices, including removal of dead plants, avoidance of end of season drought, and then application of lime and gypsum in groundnut, farmyard manure. So at harvest and post-harvest, it is important to dry the seed to 80% moisture and use of some of the resistant cultivars and cleaning the harvested pods and avoid humidification. So storage also, you have to have good fumigation. All these are important good GAPs practice. Few of the examples of GAPs, good agricultural practices that were done in management of aflatoxin. So you can see here the castor was used as a trap crop because to avoid the damage by semi-looper, which can also come on damage it. That's how we get it. You can see here the management of aflatoxin in groundnut. This particular treatment, T6, where seed treatment and then consortium application of Trichoderma and Pseudomonas, and then a neem cake application, and along with that a foliar spray, ended in number of pods being infected as 14% as against control 73, with percentage infection was 3.02, and then yield was 13, 100 pod weight was 69.54, and the kernel weight was 50. This was the kernel weight of 50.15. Similarly, in chili, the same treatment T6 with seed treatment and soil application, as well as neem cake application and a foliar spray as a pre-harvest, gave a healthy plant fruits of 1040 out of 10 plants, with aflatoxin number of fruits infected was 52 as against 292 for 10 plants, and yield was 14 quintals against 8 quintals in control. Similarly, a lot of farm trials were conducted by ICAR-ISR by Sudhini and his team. You can see here from 2013 to 2015, the pooled data indicated aflatoxin reduction percentage was 88.5. It was a detection. Seed infection was brought down by 40% and pod yield went up by 26%. So this is how it has been seen. So this is the management of mycotoxin production in maize. This is Dr. Ajit, he's an assistant professor He did his PhD also on maize toxin, aflatoxin. So this was the just last week he gave it as a package. You can see here the management using FOM enriched consortia and a foliar spray ended up in infection of 5.4% as against control of 14. Weight of the cob was almost 158 grams of the cob. Yield per plot, this was the yield, was 59 quintals per hectare with a high BC ratio of 2.92. So then we conducted farm trials in almost with the same treatments. Farm trials were conducted in 18 stations, including Raichur and Hadagali, Koppal and Gangavati. Farm trial, 18 trials, and with a large scale demonstration that indicate this is the comprehensive results of 2 years pool data in 8 stations. What we could see the end of the result, 7.42% infection in the treated plot as against 12.79 pooled results in the control with a yield of 50 quintals as against 46. This is what the package has been advocated in the last meeting, 2021. So this is what you can see, the healthy maize, and this is the infected ones. So the US Raichur has advocated last week as a package to the farmers. So this is the one aspect, Doctor, it was very near and dear to the heart of Dr. Sharma ji, where he always wanted that the PGPRs, and we have extensively used PGPRs with several traits of production of antibiotics and then formulated and used in all our studies, both in US Raichur as well as Shivamoga. So as you can see here, the basically plant growth promoting rhizobacteria, Pseudomonas fluorescens, they produce at least half a dozen of antibiotics, including 2,4-DAPG, phenazine, pyrocanin, pyrrolonitrin. They can also produce antimicrobial compound like hydrogen cyanide, siderophore productions. We got at least eight isolates of Pseudomonas fluorescens having this DAPG, which is a very broad spectrum antibiotic, and we have made use in our management studies. You can see here aflatoxin contamination, pre-harvest spray with the fluorescent Pseudomonas ended in 2.25% infection, and a reduction was 71% as against control with 8%. So you can also see here wherever Pseudomonas, fluorescent Pseudomonas, no sporulation was also observed as against control. You can see fluorescent sporulation. So bioagents as a eco-friendly were used for management. Of course, we compared with mancozeb, very low toxin fungus, fungicide. But as you can see here, the fruits infected was 13.69 in case of chili as against 24%, with healthy fruits of 16 quintals as against 8 in control, with a return of 97,000 with 1:1.85. Although as a chemical it has given good result over the bioagent, but still considering the quality chili production and export and its effect, we prefer the eco-friendly management. So resistance is one important thing, but these are post-harvest diseases. We use a pinprick method for inoculation and production of disease resistant. We could not see much of the resistance in over 100 genotypes screened, but moderately resistance could be used on breeding program. But very important there, these are senescent diseases. Lastly, I think we took up the demonstration on a large scale. This was a project funded by ICAR with two twin purposes. One is because it is an area where Hyderabad, Karnataka region, where the chili hybrids and other varieties are easily available. I call it as a green revolution technology with good irrigation, high input cost agriculture. Day in and day out farmers spray here, at least 30 to 40. So pesticide residue, aflatoxin contamination are the two twin problems where we are facing for chili. So this is what Spice Board chairman indicating higher pesticide residue and which has avoided our export to the extent of 24%. So that is why ICAR funded a project for us to go for IPM using IPM technologies. All our assembly of farmyard manure, trichoderma, then our botanicals, neem cake, and this particular pseudomonas and gourd and trap crops, all were assembled into areas, and this was the aim ICR gave us through NCIPM. So we started the project with 5 acres of land, then went into 10 acres, 25 acres, and 500 acres in farmers' field, with the actual participation of farmers. So we involved both the entomologists and pathologists on a weekly basis monitoring. As you can see here, this was a farmer's practice adjacent to our interventions. As you can see, farmer has gone in for almost 22, very conservative estimate, and we recorded what he is doing. What is the target pest? Maybe thrips or Spodoptera, whatever. So he is correspondingly using acephate, chlorpyrifos, mostly heavily loaded with chemicals and insecticides. So that's what he has ended up in 22. But this was our entomologist, pathologist, nematologist, and agronomist put together. All of them derived at interventions of amendment, bioagents, eco-friendly chemicals, soil amendments. So that is how we derived at 13 interventions. Like you can see here, our treatments, unlike farmers' practice, trichoderma, neem cake, pseudomonas, they were repeatedly being used for various target pests and diseases. And then we recorded, so for all these various diseases and pests. So this is how we use the trap crops, border crops, maintained. And then we could see the results also in integrated pest management, keeping in view. So then this adaptable IPM module was also given to different districts of Gulbarga, where it has, we could detect in the module, the fruits was 2.84 microgram per kg. In Raichur, it was around 7.58 average. And similarly, Bellary recorded 24.64 microgram. And Raichur again in another plot recorded 63 microgram when we use the adaptable module. So we also recorded, our entomologists and pathologists recorded what is the result in IPM as well as non-IPM farmers practice. So most importantly, aflatoxin contamination, at least 2% here. This was 8% as samples. Apart from mites, thrips were always lower in IPM practice, and powdery mildew was 36 as against 37 to 50. Dieback 18 to 29. Farmers practice 21 to 32. Wilt 9% 12%. And some of the natural enemies, predatory also were high in IPM practice. So when you go for economic analysis, you can see here the number of sprays were brought down to 8 in IPM practice, whereas farmers practice 22. Bioagents as many as 5. Cost of cultivation was 41,500 as against farmers practice 45,000. Dry chili was 3.5 tons as against 2.6 ton. And they got a gross return of 2.4 lakh per hectare. Net return was nearly 2 lakh, and with a benefit to cost ratio was 1.5.82, 1:5.82. So indicating, apart from getting a good monetary benefit, almost 60,000 in the IPM versus non-IPM, reduced the cost, also brought down the untold ecological damage that can happen in the agro ecosystem. That's how we celebrated the field day. So in summarizing, I think I would say that the aflatoxin is extremely toxic metabolite. B1 is the most potent. The consumption can bring in damages to liver and hepatitis B. It's vulnerable for them. Market and field contamination more common than storage. And it has affected the food chain of traders, processors, and consumers. I think we need to focus upon identifying the risk-prone area and phytosanitary measures. Good agricultural practices along with the bioagents can always bring down the contamination as per the Food Safety Authority of India. So then only we can think of consumption, storage and export and processing. So way forward, I think COVID has given a wake-up call. Whether we say it, I think food security is incomplete in the absence of biosecurity. Biosecurity, when I talk, not only human, not only animals, plants, fish, perhaps one world, one health initiative. It has transcended across the globe. So a robust biosecurity system to prevent preventive measures for the introduction of such of those devastating pests, because after all, ecosystem, it is always inherent, whether it is a man or animal or plant or fishes. So we need a very resilient food health system. If you want a resilient food health system for a safe food, a healthy tomorrow, then we need more focus on this particular research. And research is needed because we need to see the impact of COVID on crop protection and agriculture. And R&D investment, particularly on biosecurity issues. Today, as you look at here, we hardly invest about 0.3 to 0.4% of our agriculture GDP on research. Brazil 1.8%, Mexico 109%, Malaysia 1%. And the pandemic has again induced that probably the public funding, whether it is university or ICR, will dwindle for public institutions. So a policy response for, particularly in having a robust biosecurity system, in identifying, but as you see here, the black or white or these fungus require hitherto known as a weak parasites. Today they are very prominent. Are we scared? Whether we are scared, these fungi infecting on fruits and vegetables of after harvest are infecting human being. Whether there's a convergence of fungal pathogens or plant pathogens converging into human pathogens. So sustainable development goal, that's what we have been talking. Land, there is a zero hunger by 2030. We have been ambitiously waiting for 2030 to be zero hunger tolerance. Whether it is possible, we have left with another 9 years to go. If you want to really go for human development, healthy human and happy human across the globe, we really require. Of course, end of lockdown will not end the problems. This is not just the end of the impact, it is just the start of the impact. So to maximize the possibilities of agriculture, we need to demonstrate its utility and resilient system. So let us unlock the economic potential of our country. This is the everything can wait, but not agriculture. The first Prime Minister repeated, because in spite of all the difficulties last year, 1 million hectare extra planting. So that is the indication that agriculture has got that resilience to meet the requirements of sustainable food security. So we have to go along with that. Before I close, I profusely thank the research scholars, research fellows, especially Dr. Ajit Kumar, Dr. Guru Prasad, and Dr. What is his name? Yeah, 3 of our PhD scholars, and then we got projects, ICAR, ICR, and then RKVY, both the universities of my parental universities and fellow faculty who joined for this particular project. Lastly, I profusely thank the Indian Institute of Spice Research, the esteemed director, Dr. Rema, and the head of the Crop Protection Division, Dr. Santosh, and all the faculty members, and the Dr. Sarma Memorial Trust, and the family members, and paying a rich tribute to him. I profusely thank one and all for your patient listening. Thank you very much.
Thank you, sir, for enlightening us with the present status of agriculture sector in India, the challenges faced by our country during COVID pandemic, and the impact of pandemic on Indian agriculture. Your lecture on aflatoxin was very informative, and in a very simple language, you narrated the origin, the effect, and the management of aflatoxins on different crops. I am sure the talk was enjoyed by all our participants. On behalf of the organizers, we would like to thank Dr. Nayak for the memorial lecture. Thank you, sir. Now the session is open for interaction. The session is open for interaction. The participants can interact with Dr. Nayak. They can unmute themselves.
Can I talk? Dr. Thomas here.
Yes, please.
Dr. Nayak, thank you for the excellent presentation. I am from the organic sector.
Okay.
And it was very encouraging to have, all the IPM and the work that has been done in spices, especially chili. But we have an issue with use of Pseudomonas now. I think it is again being barred in most of the exporting countries of use of Pseudomonas. I think that's one thing we need to take care of in evolving the recommendations in the future. I mean, that's just my thought. It's okay to us. Thank you.
You are right, Professor Thomas. I agree with you. There has been a talk of debarring this particular Pseudomonas fluorescens, but not all species, not all strains are toxic actually. Although aeruginosa is identified as toxigenic on human beings, Dr. Andy is there. He will be able to complement and supplement you much more. But not all strains. I think they are all strain specific. We need to encourage, at least in the domestic consumptions we have been extensively using. But we need to identify whether they are toxigenic strain or not. That is the intention. Okay, sir. But if Dr. Andy has anything to add, probably I will be very glad to listen from Dr. Andy. He's the one man. Yeah.
Yes, sir. So now the current understanding is whether it is aeruginosa or fluorescence. Most of our strains turned out to be aeruginosa. The reason being our skewed selection policy. In every lab, people go for Petri plate based selection, and invariably the rhamnolipids and phenazines are produced by this aeruginosa. It happened to be the number one biocontrol agent in all screening programs. So people went with that only. So they ended up in aeruginosa. We have really come out with a clear statement like aeruginosa. There is nothing like a safe aeruginosa as on date. So it is always a dangerous organism. They can colonize like a black fungus. Dr. M.K. Nayak very clearly pointed out. Any small organism can become a very dangerous organism if you take a chance.
Yeah.
Certainly, we need to be very careful. Identification of species is very, very important. So, in my opinion, certainly we should not encourage aeruginosa for any agricultural purpose. So, this is going to be a problem in the long run. Yeah.
Yes, Dr. Andy, you are right. In fact, when we also isolated frequently, aeruginosa was dominating under our pesticide culture. But then, because of the doubt, although we did not go for strain specific, we totally avoided aeruginosa from IPM or what strategy.
Yeah,
There's a fight, Professor Thomas. Yes.
Thank you. But the bigger problem is at the ground level, people doesn't understand it. And
Exactly.
And with all the the government schemes in promoting bioagents, even you can even have it in your courtyard with a pressure cooker. That's the culture of bioagents being promoted, which is very, very devastating in the long run. I mean, I think we need to take a very good stand on that side. Thank you.
Sir, Thomas, sir, to add one more point, because a good audience is sitting here. The selection policy, of course, you can go for plate-based selection, no doubt. You can bring one more selection policy in your own laboratory, the temperature tolerance of those isolates. So you put a 37 as a criteria. If something grows on 37, don't take it on board for any future purpose. because human body temperature around 36.8, if something grows at 37 is going to be a problem for you as well. So this is one of the criteria I think typically need to be followed by individual labs. So there are Pseudomonas, they do not grow beyond 30 degrees. So those organisms are very effective biocontrol agents. The Pseudomonas fluorescens included here, because they never grow beyond 35. So my own experience. So they grow better at 25 to 28. And those organisms are very ideal for all our organic agriculture or any kind of biocontrol programs. This small intervention can help, sir, to avoid later problems.
Yeah, you are absolutely right, Doctor. 37 and above, perhaps we should discourage.
Exactly.
Yeah.
Sir, I have one question. This is with regard to our future strategy. Now, are you visualizing that anything which maybe a convergence of medical and the agriculture scientists coming together in dealing with such pathogens?
That's what I foresee. I foresee because medical mycologists definitely they have their own agenda. Definitely they are working out for how to make yourself, whether it is aspergillosis or pulmonary infection or lung infection, to make you free. But right now, if you look at the way the black fungus is treated, initially they go with these medicines, and that is not the end actually. Unless that particular fungal ball is removed from the eyes, he is not safe, and even that is also doubtful. So that's why I see one of our scientists, employees' father, who has been in the hospital for the 18 days. He has not come out of the black fungus, but then the doctor has also told that he has to be operated. So, but his body is not in a position to accept the surgery right now. What I mean to say is there is a lot to do by mycologists, fungal pathologists, in understanding many of these, infections, their predisposing conditions. We were just, keeping on the dining table the cut fruits, but we used to find rhizophus, some of these molds growing, but we cannot neglect now. That's what I mean to say is probably we have to understand the entire whole profile of these organisms, their survival, their proliferation. And yesterday's newspaper, when I was scanning, there was a news item from Bangalore Medical College saying that the person has not been administered this steroid, and he has a COVID, fine. He has got the infection of black cap, because you can always say that the COVID has made him immunocompromised. But then there is another person without any COVID, no steroid, but he has got the black scars. So these are some of the frightening, although I don't want to make all of us scared here. But then there is a lot of things for the medical mycologists and plant pathologists to sit together and work out, because biosecurity, your food security will be incomplete in the absence of biosecurity. We can't say that your food is safe. So as I understand, 142,000 people die every day because of the food illness. Food illness, that is account of 24% of the death, total death. World over, I think many of those developed world are very cautious in food safety. So India, we are in a stage of food security, turning towards nutritional security. Now again, this COVID has pushed us back. So that is why I think the bigger vision for understanding all these fungi, even including alternaria, they are all, lung infection. They can always bring some kind of allergies, aspergillosis. So I think there is a big think tank needed in identifying and having a network of all these people working together, medical as well as where they come from, whether they're plant product. After all, they come from plant products. So this has to be understood.
Do you think? Why do you think the prevalence of black fungus incidents are more in India after post-COVID?
Tropical climate.
I think
The tropical climate.
tropical climate, humid weather,
Exactly.
pollution is very quick. And we have also a reason to believe that we waste so much of fruits and vegetables just on the veranda. We don't have a sanitation for those things. They can easily come and occupy on the surface and sporulate very quickly. Now, monsoon has started. You can see the fungal flora in the aerobiology. You can see their density very high, very high. Even aspergillus, very high. That is the reason I told 0.14%. That is the number of COVID, mucus black scurf cases, which is 80% more than the developed world. Statistics says, statistics reveals.
May I speak? Hello.
Yes, sir.
Yes, sir.
Yes, Namaskar, Dr. Naik. I am Dr. Mathur.
Oh, great.
Oil
Great
palm.
to see you. Oil palm.
Yes, sir. I was very happy to listen your talk.
I was excited to see you here.
But then I am recalling my days spent in National Research Center for Groundnut.
Yeah.
Junagadh. I was there for 7
Yeah.
years.
Yeah.
And there we also were bothering for this aflatoxin. It
Yeah.
is a problem in groundnut where EPIDA approached at that time, while 2 consignments were rejected for the having high aflatoxin levels in groundnut, which were exported.
Yeah.
And if you see the levels which they are asking in exported material, it is very low. It is very low. Sometimes it is very difficult to achieve also.
Yeah.
And that time EPIDA approached us what to do and how to do. That time only I came across one process or one technology from Australia where A. flavus strain, which is virulent and causing this aflatoxin infection, there was a virulent strain also, which is virulent on this strain, which is causing the aflatoxin.
Yeah.
So whether that approach is correct, since I just now listened Mr. Kumar, that we should not advocate any strain or any biocontrol, what I understood, which is about 37 degrees Celsius. They are active like that. But then most of our area is high temperature areas. So whether they will be effective in controlling that sort of problem through management.
Yeah, actually, sir, Dr. Mathur, this was debated also in many forums.
Yes.
Although we say ambient temperature is above 35 in tropical countries, when it comes to the plant associated niche where the diseases are coming, the temperature is, there is an ambient temperature is always there. For example, it is anywhere between 25 to maybe 32. It never goes beyond that actually in a micro niches, right? Where what we call infection court in plant pathology. So there the temperature seldom goes beyond 32. So there comes the microclimatic conditions are quite different from the macroclimatic things. So I think it's not a correct, a discussion where we have an ambient temperature of around 37 and we have a completely different kind of climatic conditions in a micro niche in a plant where the disease comes. So these two are two separate things, sir. So from that perspective, certainly we cannot encourage something which is growing at a higher temperature, especially the 37 was a very critical point. So actually, in fact, this is implemented in some of the laboratories abroad. They routinely do it actually, not to encourage certain organisms which eventually will be rejected at the end of the day. So they nip the problem very early on so that good strains with all safety things are taken on board for any kind of commercial purpose. I think this is one thing we need to really implement because our biodiversity is so enormous. There are organisms, the mesophilic ones, having an excellent biocontrol potential are still around. So our skewed selection policy right in the particulate level is create a little bit of problem for all of us. So including our own group, we most of the time we end up in something which is not so good for safety purpose. So we need to be very careful, sir, in the selection policy. The best way forward is you try to include the plant, whatever may be the plant, in the selection process. Now, to add one more point, with the permission of the organizers, now one more screening is slowly gaining popularity. What is called as the innate immunity contributors. For example, certain strains, they can induce the innate immunity in plants. So certainly this protocol necessitates somebody need to use the seedlings of any plants where they are going to use these organisms as a biocontrol agents. They can very well use those seedlings in the screening process. And you select those organisms, they are able to induce the innate immunity against the so-called pathogens. So the in-plant assay, which is going to tell you whether the organism can induce the innate immunity in plants, in addition to the antibiotics, whatever we are routinely doing, that would do a better job. So that end of the day, you have something very precisely modifying the plant, if not permanently, at least transiently, so that the disease is not really causing economic damage. So antibiotics plus induced defense can become a wonderful combination. Yes, sir. Thank you.
Thank you.
Dr. Mathur,
Yes, sir.
the one which you are talking about, atoxygenic one, infected, toxigenic, and giving a biocontrol. Literature is there on ECRISAT also, they have done some work, but again, as Dr. Andy put it, it's a question of safety then. We don't know if there is a mutation or any other recombination, something like that.
Yeah. Horizontal gene transfer, yeah.
Yes,
That's another threat.
And data that I provided also indicates sometimes you couldn't find a gene there, but there's the elaboration of toxin. So these are some of the things like our RT-PCR showing false positive, false negative. You should be very careful about these things.
Okay, thank you. And thank you, Dr. Kumar. What you told is a new for me.
Yes,
so
sir.
I will discuss separately with you. Thank you.
Sure, sir. It is gaining popularity, sir, now. Yeah.
Some of the isolates of streptomyces, they have been identified to be, because they are known to be having production of antibiotics and induced systemic resistance also. But then the same limitations come here also. How long? How safe? This is the problem.
Any more questions?
I will be very glad if you can mail your questions, because concurrently there is a session for me in the NAS going on. So I have taken permission. Kindly, you please send your questions to email, or maybe you can send it to organizers also. We can have a discussion. And please permit me to join the other session. Right.
Thank you, sir. So we are about to conclude the 5th Dr. Vaya Sharma Memorial Lecture. Now, may I now request Dr. Anuradha Yamanaru, daughter of Dr. Sharma, presently working as scientist with the Moderna Labs in Boston, USA, to deliver the formal vote of thanks.
So everyone, it's a privilege to be part of this 5th annual memorial lecture in honor of our dad, Dr. Vaya Sharma, who served Indian Institute of Spices Research from 1975 until 2002. Little would Dr. Sharma have known that this institute, which he served with devotion, and the farming community he so much loved and respected, would keep his memory alive by forming a trust to enable realizing some of his dreams, which include supporting the farming and scientific community. The love and respect we have seen being poured in these meetings touches our heart, and I believe will touch his soul wherever he is. In line with one of the goals of the endowment fund, which is to enrich the knowledge of the scientific community on various topics, it is a pleasure to have the Honorable Vice Chancellor, Professor Dr. M.K. Naik, to deliver this year's memorial lecture. I did not know enough of the professional connection that you had, sir, with Dr. Sharma, though I definitely remember that he was associated with agricultural universities in Karnataka and toured places like Shimoga and Dharwad for his work. I'm happy to hear your association with our dad, and thank you immensely for paying your tribute to him by reminiscing his contributions. Sir, on behalf of the endowment and on behalf of Dr. Sharma's family, I extend my heartfelt thanks to you for a very informative talk on a very relevant and informative topic on the effect of mycotoxins during this pandemic on agriculture. It is very interesting to know the role of fungi that affect crops in possibly causing mucormycosis, or the black fungus infection that we are seeing in many patients post-COVID due to compromised immunity. It was also very informative to learn more about how mycotoxins cause food contamination, human disease, and losses in crop production, and consequently the economy. It was nice to see your work on aflatoxins and the use of GIS mapping and low-cost culture methods in detection of mycotoxin contaminants in produce. It was also nice to hear the practices highlighting the use of microbial agents and IPM that Dr. Sharma contributed immensely to manage aflatoxin levels. As you mentioned, population is a real big issue, which has put stress on all the resources, especially in these tough times, resulting in distress and hunger in the less privileged communities due to mycotoxins. With the interactions that we have seen today, attendees have for sure benefited from your talk. This makes the organizers more happy about these lectures. On a lighter note, sir, I would like to let you know that one of your students is one of my best friends here in the US. Uma and her husband Chandrasekhar, I do not know if you remember them, have been your students years ago, and they are big fans of your teaching. So they have asked me to convey their best wishes to you today.
I very well remember her. In fact, of course, recently I did not talk to her. Please convey my regards.
Sure.
And thank you very much. Right.
I once again thank you, sir, to grace today's event and speaking on a very useful topic. Next, I would like to thank Dr. Rama, Acting Director, IISR, for all your support for this endowment fund. Thank you for your tribute to Dr. Sharma with your generous, kind words narrating Dad's vision. Dr. Rama, I would like to take this opportunity to express my pride to see a woman heading IISR. I congratulate you for taking up this responsible role as the director of the institute you have served for many years. We wish you all the girl power to give your best to make impactful contributions in your new role. We look forward to working with you for the Endowment Trust in the coming years. Dr. Santosh Ipan, it has been a pleasure to have you as the driver of this trust from its inception 5 years ago. I have always known you as a very smart scientist and a very organized and creative person. These traits have immensely helped the trust to shape its structure and goals. Being the head of the plant protection group of the Institute, which my father was part of, you have been the force behind organizing every lecture in the past many years. These lectures have been very well received by the audience and hopefully have benefited them. Undoubtedly, again, I'm sure You have played an important role in launching the official website for the Trust, and we congratulate you for all your efforts in this regard. We hope to work with you for years to come for the Trust. Dr. Nirmal Babu, you have been a solid support for this trust as one of the founding members who took the initiative to form this endowment fund. It has been a pleasure to see your affection and respect for Dr. Sharma during and after his term on this earth. Sir, I hope you are thoroughly enjoying your retired life. Not to put any pressure, but the person I have known you to be, I'm really looking forward to seeing you fulfill a wish you have with regard to a project, which I'm sure you're working on. I would leave it to you to guess what I'm alluding to. I would like to take this opportunity to thank each and everyone who has supported this trust with their contributions. It means a lot to us. It shows your respect and love for Dr. Sharma and your hope to be a part of something noble in service of the farming and scientific community. On a personal note, we, the family of Dr. Sharma, would like to take this opportunity to personally thank several people who I'm hoping are present here. Dr. K.R.K. Reddy, Dr. M.S. Reddy, Dr. Narsimha Reddy, Dr. Sushila Bai, Dr. Champakam, Mr. Radhakrishnan, Mr. Shashi, Mr. Ahmad Kuti, Mr. Mayan, Mr. Yohanan, Mr. Kumar, and many, many others who I have not mentioned, who have shown immense love to our dad in terms of giving company when he was alive. I want you all to know that every visit you made, every phone call you made touched his soul, and he so looked forward to spending time with all of you. Being far away from us, his daughters, it would have felt that he was lonely. But no, with your company, he was so happy to be home at Calicut among friends, because friendship is what he always treasured and considered very important in life. I also thank Dr. A. Jeevalata, who is the co-convener of this event, for all her efforts in coordinating this lecture. I also extend our immense appreciation to Praveena for emceeing this event. Lastly, but not least, I would like to say thank you to every scientist and supporting staff and all the students that have in one way or the other supported Dr. Sharma during his time at IISR and outside. Each and every one of you have been an important part of his life. And thanks to everyone on this WebEx meeting for making time to attend this lecture and participating in a very useful discussion, sharing your experience with others. The endowment hopes to bring more such events to support the scientific and farming community in the future. Before ending, I would like to request all of you to spread the word about this trust so that it grows to be stronger, to give back to the community. Thank you, one and all. Jai Hind.
Thank you, Anu ma'am. So we came to the end of today's program. On behalf of the organizers, we extend our sincere gratitude to all the participants.